sponsored by Cytiva have managed to publish or disseminate and generate as part of their research activity. So I always start off with this sort of side of the research rationale, because I think it's important to understand the context of where the research I'm leading is coming from and kind of why we do what we do. And it really comes down to, one of three key reasons. The first is the challenges that we're seeing with manufacture. So these are just kind of some headlines that we've seen over the last couple of years when it comes to cell and gene therapy manufacture, which, these therapies are transformative, they're curative in many cases, and they can have a significant positive impact on patients, but the manufacture remains a challenge. So here, for example, we know that Bluebird had a challenge with their Zynteglo product in terms of the manufacturing aspects, and that tripping up the development and the launch of that particular product. Likewise, that's not limited to Bluebird, but we've seen that across the industry, and more recently, BMS had a major challenge with the delivery of their CAR-T Abecma product. So we're starting to see that although we can address some of the issues around efficacy and safety, one of the key challenges that remains, especially in terms of patient access and cost, is that of manufacturing capability and bioprocess development. And that's not something that's just recognized by the sector or the industry, but it's also something that has been picked up by the regulator. So Peter Marks, the CBER director within the FDA, highlighted that actually one of the key challenges cell and gene therapies face is the sustainability and the commercialization effectively and the accessibility of these therapies, and linked it fundamentally to the manufacturer, and highlighted in particular that automated manufacturing could be another solution. And that builds on previous statements from previous FDA directors who highlighted that we need to develop scalable manufacturing technologies if we're going to sustain and develop these therapies moving forward. And that's fundamentally the core of the research program that I lead. Often one of the challenges we had with cell and gene therapies is that we have variable input coming in, but we still have to deliver a consistent output. And I remember when I first joined the field about 10 years ago, I was an undergrad at UCL and inspired by Professor Chris Mason. And one of the challenges at the time was there were very few products, if any at all. I think there were some tissue engineering products which had been approved by the regulator. But the challenge was, how do we get I remember the kind of consensus, or the main challenge within the sector was, how can we get these therapies approved by the regulator? Fast forward maybe 10 to 15 years, and it's fantastic to see we've now got tens of products approved by global regulators, the US FDA, the UK MHRA, the European EMA. So we've got a number of products now approved by regulators, and for me, the fundamental challenge we now face, which and I say this as potentially as an existential threat to cell and gene therapy moving forward, is we have to be able to demonstrate commercial viability of the sector. It's one thing being able to develop these products. It's another thing entirely being able to develop them at a cost that the market is willing to pay, that it can afford, and importantly, how can we start to ensure worldwide patient accessibility, not just for the developed world, but how can we make this more accessible to patients in the developing world? So as part of that drive for commercial sustainability and viability, a lot of the work we've done addresses a lot of these key issues, trying to improve manufacturing processes so that we can bring the cost of these therapies down, improve some of the aspects around the business modeling and cost of goods, and that's in collaboration with my colleague, Professor Suzanne Farid, who's one of the world-leading experts within this space, and starting to think about how we develop small-scale models and implement things like quality by design, which is the status quo, particularly in biopharma production. In terms of the research that my group leads, so we have activities within the adherent suspension stem cell space, so things like mesenchymal stem cells, hematopoietic stem cells. We have extensive amount of work in the immunotherapy or immune cell space, so things like T lymphocytes, natural killer cells, and so on. And then over the last 3 or 4 years, a big focus in gene delivery, both lentiviral and AAV gene delivery for gene-modified and gene therapies, as well as a big focus on non-viral gene delivery. So looking at things like electroporation, mRNA, as well as delivery using things like lipid nanoparticles. And the work I'll be touching on in this presentation will kind of focus on all of these key areas. So when, the question that myself and our research group tries to focus on is, what does the future of manufacture, or what does the future of cell and gene therapy manufacture look like? What will it look like now, or what does it look like now, and what will it look like in 5, 10 years' time? And that's kind of one of the things that I always try and revisit and reflect on as the sector advances. And I've tried to kind of break it down into 3 key areas that we've identified and that I've tried to align my research to. So the first is kind of automation and this concept of decentralized or point-of-care manufacture. The second is looking at how we can optimize bioprocesses and ultimately control for improved outcomes and efficiencies. And then finally, how we can start to use digitalization, so digital tools such as AI and machine learning. And that's done extensively with a number of collaborations internally within the department with various colleagues, as well as with external industry partners. So starting with kind of the case study 1, the automation strategies and decentralized point-of-care manufacture. The first kind of focus on automation emerged when I'd done my PhD, and my PhD was focused up on, how can we scale up production of human mesenchymal stem cells? And this was back in kind of 2012, 2013. How can we scale up stem cell production? So adherent cells, which are sticking to microcarriers, how do we scale up that production? And we got to the 5-liter scale. And then I realized actually from a research and development perspective, it's probably more valuable to be able to develop a small scale model. So rather than going up to the 5-liter, let's go down to the 15 milliliter scale and develop a high throughput model that can enable us to kind of run extensive experiments and generate significant data sets. Because I remember, and this may be a shock to many of you who work with CHO cultures or E. coli, a bottle of, let's say human embryonic or mesenchymal stem cell medium can go up to 500 pounds, Great British pounds. How much that's worth now, I don't know, but 500 pounds per 500 milliliters. And so we're looking at very expensive media, which from a research and development standpoint is clearly not sustainable for academic labs in particular, but also becomes a challenge for industry R&D. So could we develop a process that could scale the process rather than go up to 5-liter down to the 15 milliliter and be able to demonstrate similar outcomes and conclusions? And the answer was very much yes. So through a partnership with one of our industrial partners, we could demonstrate, we could generate, using the AMBER-15, an effective small-scale model for human mesenchymal stem cell production. And that was my first foray into kind of automated manufacture, the idea that you could have robotic systems running processes and so on. That then extended into looking at how we can then apply that, because of the success of that project, to other platforms. And there was a larger platform called the AMBER-250 at the 250 milliliter scale. How could we apply it to there? And part of that work, and again, this was a successful project we led at UCL, looking at how we could develop a new vessel to support or improve production of cell and gene therapies, which are often adherent or need some bead-based culture. And so we developed a new vessel in partnership with the industrial partners to look at how we can improve production and generate effective outcomes. And that vessel is now being marketed and sold by that particular company based on the research that we had undertaken. And we published kind of a number of papers off of that work as well. But that work was pivotal for what was to come next. So we'd established that work with human mesenchymal stem cells and microcarriers. And at the time, I had a fantastic NGD student, Elena, who said, well, actually, seeing as we're developing this new bioreactor, how about we apply this to CAR T cells? And CAR T cells are suspension cells, so they should grow in most tank bioreactors, but they weren't very good at growing, and I'll show some of that data a bit later. But one of the key challenges with CAR T cells is that they have to be activated, and the way they're activated is often using beads. And so as some of the work we'll show a bit later, we noticed that the cells were not being activated, they weren't coming into contact with the beads in the original vessel, but this new and improved vessel led to significant improved outcomes. But then the third aspect of our kind of automation pipeline was a large European project working with a number of key partners, particularly in Germany and in Ireland, as part of the €6 million AutoStem project to develop an automated manufacturing platform for stem cell production. And as part of that, the idea was to kind of develop a process where we could take cells from a donor and have the entire process completely automated, right the way through from isolation of the material through to production in the bioreactor and then delivery back to the patient. And I'll be honest, when the first schematic was launched of the platform by our colleagues at IPT Fraunhofer, me kind of being my usual British pessimistic self, thinking we've got 3 years and €6 million to develop this program. We're probably not going to get there. And I'm delighted that, throughout the course of the project, we had some fantastic partners, and I was delighted to be proven wrong, because although this was the initial schematic, we were able to develop the platform, develop a completely validated GMP, or I say GMP-like, it wasn't GMP, but under GMP amenable conditions, and demonstrate that we could effectively grow mesenchymal stem cells on microcarriers in a stirred tank bioreactor within the platform, and get equivalent results to a manual bioreactor process here in the UK. This is some of the data that's been published from those studies, comparing the automated versus the manual process, so the automated in blue and the manual in the light orange color. And this has been published, as I mentioned. And I think the proof is normally in the pudding, when you can kind of see things visually. And here's a kind of a brief video that showcases some of that technology in operation. So here was the platform at the end of a process run. So we've expanded the cells in the bioreactor. We're now harvesting the cells, and this is undertaking the cryo work. So this is now going to be transferring the material into these cryo vials and then banking them down. Now, for me, this isn't particularly innovative and novel and exciting. It's very nice to see robots, but robots have existed for decades. And it's simply replacing what a human could do. But what we were trying to do with this proof of concept platform was show that actually we could get to the point where we could potentially manufacture multiple patient cells in the same system in a completely segregated format, which is one of the key limitations in current manufacturing platforms, where once you get the patient cells in the expansion system, it can only be used for that patient. And in many ways, you have an expensive incubator for 10 to 14 days, and you can't use it for any other process. And what we're able to do in this system is have multiple bioreactors running in parallel through segregated systems, and that was done in conjunction with conversations with the EMA. And so although this was not a completely GMP run, it was run under what we call GMP-like conditions, fully validated and sterilized from that perspective. And I like to show this point here, because whilst it's completing the cryopreservation for one patient, it was running another bioreactor run for another set of donor cells. And so it's just going to do what a human would do. And for those of you that have used cool cells in the past, one of the biggest challenges is once you've added all of your cryo vials, the challenge with the cool cell is actually getting the lid back on for any operators that have used it in the past. And we had to come up with, actually, that took a few weeks with our German counterparts to kind of get the right pressures and angles to be able to get the cool cell lid back onto the device itself. So the way it does it, it puts it on gently, and then it gives a gentle tap at the end, as you can see there. So I won't show the rest of that video. So that kind of covers a lot of the automation work that we've done. And we've now, as I'll show at the end of this talk, extended that project where we've got additional funding to focus on CAR-T point-of-care manufacturing. We're making significant progress in that regard. But from my perspective, for cell and gene therapies, whilst not everything will have to be point-of-care or decentralized, it certainly will be very useful and maybe important for certain types of therapies where the material either can't be cryopreserved or patient vein-to-vein time is of absolute critical or is absolutely critical to patient outcomes. And so as part of that, we've been involved in leading a number of kind of editorials and kind of position papers focusing on this concept of decentralized manufacture. I won't go into that today, but that's been a key focus of the work that we've been doing. And that's led to, in part, some of the work that the MHRA have led on. So the MHRA's Innovation Office, led by Jan Ries, focused on delivering a point-of-care framework for advanced therapies and more broadly healthcare therapies as a whole. But advanced therapy was a core focus. And that was driven by the fact that about 5, 6, 7 years ago, we were having these conversations, and I actually attended some of the early MHRA stakeholder meetings. And that, obviously, the work that we've done has led into that, and it's been fantastic to contribute directly to some of the regulatory outcomes in the point-of-care space. So the second case study then focuses on bioprocess optimization and control for cell and gene therapies. And so I have a few slides, but I'll try and kind of run through them just to give you a flavor of what we've done. So really, there's kind of 2 areas of focus for the work that we've done. One is in the viral vector optimization space, both for lentivirus and AAV, and I'll touch briefly on some of the lentivirus work, but also in the gene-modified immunotherapy space, so particularly with T cells and CAR T cells. So this is work undertaken by one of my doctoral students who's now I am proud to announce that I can now call her a doctor. She's passed minor corrections, and I believe she submitted those corrections. So many congratulations to Karma. So Karma, who led on and was funded as part of the UCL-Cytiva Centre of Excellence, led on some work to look at how we could generate improved lentiviral vector outcomes that were more industry ready, avoided the use of serum-based media in industrial platforms. And her work was to look at how we could generate lentiviral vectors in the iCELLis Nano platform for adherent HEC293 T cells. So the focus of this was very much kind of looking at, again, with the CAR-T, CAR-NK space in mind, but how could we generate viral vectors at a lower cost, ultimately? Because although CAR-Ts are fantastic, we know they're very expensive, and a large proportion of the cost is down to the viral vector that we're using. So could we bring those costs down? And so the focus was to develop a scalable and robust upstream process for chemically defined production of lentiviral vectors using adherent HEC293 T cells. And so the first part of her work was to look at how we can do this at the small scale. So looking at a lot of the process parameters, particularly supplements, things like sodium butyrate and other aspects, as well as looking at the removal of serum from the process and moving towards a chemically defined media. And in a nutshell, we could show that we could improve the bioprocess and move away from serum to a chemically defined medium in T flasks. But the question is, could we apply that then to a perfusion-based adherent process in the iCELLis Nano, a more scalable platform for the production of adherent HEC293 T cells and therefore lentiviral vectors? And obviously, the great thing about the iCELLis Nano is the fact that there is a scalable pathway from the Nano to the larger iCELLis system, which is used extensively within the sector. And what we could show is effectively that our freestyle 293 medium, which was our serum-free process, was able to generate significant amounts of the virus within the iCELLis Nano, and actually slightly higher than our serum-based process, which was kind of the gold standard at the time. But the question then is, could that virus that we're generating, obviously we've got kind of end goals in mind to deliver CAR T cells and CAR-NK cells, so we have to use the lentiviral vector to deliver a gene of interest, in this case anti-CD19 CAR. Could we deliver that to the T cells and the NK cells and still have those cells generate efficacy outcomes? And the answer again was very much yes. So we published the first piece of work. We're in the process now of publishing the second piece of work showing the effectiveness of the virus to transduce the T cells and the NK cells to generate CAR-T and CAR-NK. And what we showed was that the T cells were fantastic. Obviously, this particular CAR construct, the anti-CD19, is designed for T cells. So it perfectly transduced the T cells. And although it transduced the NK cells and the NK cells were efficacious, they didn't have as much efficacy as the CAR T cells. But that was to be expected because, again, this CAR construct was designed for T cells. But in both cases, the NK cells and the T cells, once they were transduced with the CAR, were able to target and kill the cancer cells. So that for us was a huge success and kind of a key milestone and something that we're very keen to publish as well. So that kind of focuses on the vector side. Now coming to the actual human T cell and CAR T side. Another key part of the work is how do we establish scalable production platforms for CAR T therapy production? And so some of the key research questions focus on what are the key culture parameters? Basic research questions, but we felt these were critical because they hadn't really been addressed in the literature that we had seen about 5 or 6 years ago. And I would still argue they're probably still not fully quantified and fully explained. That was one of the key questions. The other question we had was how can we control T cell and CAR T production processes to improve final product yield and quality? And then finally, what is the impact of scale and manufacturing platform for T cell manufacture? Because when you look at it, and these are just the platforms we have in our labs, there are a number of platforms that are used both in literature and commercially to grow T cells and CAR T cells. Some of you may laugh and look at T flask culture bags and so on. That's probably certainly R&D academic. There are actually processes which have gone through phase 1 and phase 2 still using culture bags. And that just shows the rudimentary nature of some of these manufacturing platforms. I joke about T flasks, but again, they're used certainly in phase 1, phase 2 clinical trials by academic or clinical counterparts, which presents a major challenge when we start to think about scalability, commercialization, and so on. So our focus is how can we use some of the more automated platforms or scalable platforms to try and generate improved yields, but also improved efficiencies and integrate things like process monitoring and control. And a key focus for us has been starting to look at stirred tank bioreactors. So one of our key basic research questions, and this, as I mentioned, started with Elena back in kind of 2017, 2018, was, could we grow T cells in stirred tank bioreactors? It sounds like a very basic, easy question. It is a basic question, but it's not particularly easy, I guarantee you. And one of the reasons we felt it wasn't easy was that all of our clinical counterparts at the time were, questioning, why do you want to grow T cells in stirred tanks? These are cells, although they're in suspension in our bodies and so on, they don't like to be agitated. They don't like to be disturbed. You need to grow them in flat surfaces, which is slightly odd because they were being grown in the wave bioreactors developed by Cytiva. But also, when you compare the shear stresses that the cells are experiencing in our blood, in the bloodstream, one of the key concerns was shear on T cells because of the stirred tank. It would actually be higher in our blood, especially individuals that have higher blood pressures, than they would be experiencing in some of the stirred tank bioreactors that we're going to be using. But, and I like to show this slide because when we did our first studies, and I was excited when Elena said, "Oh, I've just started running the T cells in the spinner flasks," we actually found that the T flasks were very good and they were generating what we'd expect. The cells in our spinner flasks were dying, and it made me think, maybe these clinicians and academic colleagues and biologists and immunologists have a point. Maybe there's a reason why we can't grow them. But then it suddenly dawned on me. There were kind of 2 or 3 key observations at the time. I made earlier the point about the fact that we have to activate T cells. So T cells, we have to mimic what happens in the body when a viral or bacterial cell encounters a T cell. It elicits a range of immunological responses. The T cells become activated, and that causes them to grow and start to expand and so on and so forth. And the way we mimic that in vitro is we give them these dynabeads. So these dynabeads have certain antibodies which are coated onto these beads, and they come into contact with the cells, and that kind of mimics that response. And what we found was we were using spinner flasks, which are magnetically driven agitators, and these dynabeads were also magnetic. So the dynabeads were all sticking to the center of the impeller, and were not coming into contact with the cells. And that, that was visually observable. But it made us think, actually, and that led to some important work later about the importance of bead contact, or the importance of activation generally, but also importantly, bead contact with the cells. So that was so we thought, okay, let's move away from the spinner flask. Let's move it into a small scale stirred tank. So we moved it into the AMBER 250. And what we showed was that we were able to, unsurprisingly, generate excellent outcomes when we moved them into stirred tanks at scale, and particularly when we took them to the higher agitation rate, surprisingly enough. So in spite of all of our clinical colleagues saying actually they're going to die and you're not going to be able to grow them, the higher the agitation, the better the cells grew. And that, again, fundamentally reaffirms our hypothesis, the importance of the cell-to-cell, bead-to-bead contact. So that was with primary human T cells. We showed we could grow those cells and they could grow well. The next question was, could we grow CAR T cells in stirred tanks? So cells that have now been transduced with CAR. And the answer again was very much yes, and we actually took it even higher. So in the previous study, I think we went up to 200 or 250 revolutions per minute. We actually took these up to 500 revolutions per minute, and we had identical outcomes. And what you'll find interesting is actually at our lowest agitation rate, the 100 RPM, where we had the cells that didn't grow very well at all. And again, reaffirming our hypothesis, because some of the beads were in suspension, some of the beads were not in suspension, whereas at 200 RPM and above, once the beads were in suspension, they were fully in suspension, and we had equivalent outcomes between 200, 300, 400, and 500 RPM. Now, we opted to move forward with 200 RPM and the associated energy dissipation rates for future scaling studies, but it's useful to know that we can scale beyond, in this case, up to 500 RPM and maybe beyond. We haven't looked, because as you start to get to mass transfer limitations at scale, one of the main ways you try and avoid mass transfer limitations in the first instance is to increase the agitation rate to improve the mass transfer. Now, we don't need to do that at this stage, but it's good to know that we've got that flexibility and leeway to be able to do that a bit later. The next question then was, okay, we've shown we can do it with T cells, we've shown we can do it with CAR T cells. can we now do this at the litre scale? So we've done this at 250 millilitres, and the answer was very much yes. And actually, more importantly, not only could we do this at the litre scale, but we showed right away from the 15 millilitre model through to the 250 millilitre model and up to the 1 litre model, we had effectively equivalent outcomes. So we're starting to show, and we have been able to show, equivalency in terms of a scalable process right the way from 15 millilitres up to 1 litre. And we're actually now beyond the 1 litre. We're actually running studies as we speak, or maybe not as we speak, I think as of last week, running studies that are going well beyond the 1 litre. And that's a key focus for us to try and establish an end-to-end process. But that's been kind of one of the key outcomes of the last couple of years, is to demonstrate scalability moving forward. But those were kind of the, proof of concept. Can we do this? Can we do that? And can we grow these in stirred tanks? Now that we've demonstrated that, the next question is, how do we optimize that? So the benefits of stirred tanks, or one of the key benefits, is not only the scalability, but actually the process control that comes with process monitoring and control that comes with the stirred tank. So being able to measure more parameters, being able to control more parameters, and would that lead to improved optimization? So we had another research question building on this. Could we establish process intensification methods to reduce process time or increase yield? And the answer very much again is yes. So I won't go through all the lines on the graph here, and we are looking to publish this. We've been sitting on this for a while for a couple of reasons, but we're now in this position where we want to publish this and get this out there, where we've been able to show from, if you compare the purple line and the brown line here, that's probably the best example. These are identical media, identical donors, identical processes in every way, except for the fact that in the brown line, we are now using effective process control. So rather than feeding at set time points, which is the usual process that many undertake for T cell production, we're now dynamically feeding the cells based on certain things that we've been able to measure, both offline, but more importantly, at line and online. And with that kind of dynamic feeding regime, we've been able to generate effectively the same number of cells that we have with our standard process, but in almost half the amount of time. So going from a 7-day process down to a 4-day process. And we've actually brought this much lower, and we're now working with colleagues and hoping to put in a couple of grants with colleagues such as Claire Roddy, who's one of the leading UK clinicians on CAR-T, to look at how we develop a 24 or 48-hour manufacturing process for CAR-T. So we showed that we could, through effective process control, we could grow the cells and so on, but expansion is one thing. The other question is quality. Do the cells retain the in vitro and in vivo characteristics that we expect? Effectively, especially with CAR-T cells, do they home in and kill cancer cells? And so this particular video, or these two videos, I love to show, because the first on the left here shows T cells and cancer cells. So the normal T cells, these haven't got CAR, so they're not able to recognize the cancer. The T cells are in gray and the cancer cells are in green. And what you'll find is that over a period of 24 to 48 hours, the T cells can't recognize the cancer cells, and the cancer cells continue to grow and grow and grow. Even though there are lots of cancer cells and they continue to grow, the T cells are not able to hone in and kill the cancer cells, which are labeled in green. However, when we transduce the cells with CAR, and obviously these are cells that have been expanded and produced post-harvest in a stirred tank bioreactor, do they retain their functionality? And the answer is very much yes. So we have exactly the same assay. Again, we have the T cells, but they're now being transduced with CAR, also in gray, and our cancer cells once again in green. And what you'll find is over the same time period, the T cells or the CAR T cells are able to recognize the cancer cells, home in on, and then kill the cancer cells. So over the same time period of 2 days and 20 hours, on the left-hand side with our T cells, we've got a culture that's effectively green, with full of cancer cells. And on the right-hand side, we have very little, if any at all, of the target cancer cells remaining, because the CAR T cells are able to identify and subsequently kill the cancer cells. So one of our final research questions, so there's kind of two more aspects to this. Could we establish a process that is adaptive to individual patients? And why is that important? Because in a standard process, we have fixed control. So we'll have a fixed control process where the material will be going through, maybe certain time points that will be fed or whatever it might be. But that's clearly not conducive to patient material, where with every single patient, the input is going to be different. With every patient being different, you may have to alter the process slightly, but you can't do that with a fixed control process. And under traditional bioprocessing or regulatory approved products, you can't generally do that. You have to have some sort of fixed control process. But what often that leads to is a variable outcome, because you're not able to change your control parameters to accommodate the incoming variability. However, the idea of adapting our control process to the incoming patient material would, in theory, lead to a more consistent output, or that being the idea. So if we can control the process or adapt the process to the adapting variability that we get with the patients, that would hopefully lead to a more consistent outcome. And the way we can do that is by, obviously, with the starting material, we can measure certain things and we can adjust certain parameters. And that's been kind of what we focused on in the last kind of 12 to 18 months. Can we develop these adaptive manufacturing processes? And here's an example of some of the most exciting data we've had. And again, we're looking to publish this based on, we've held it back for a couple of reasons, but we're looking to publish this very soon now that we've been given the green light for certain things. Because why this data is so exciting is it's all from the same donor material. So this was an individual over the age of 60 and what we'd call in our labs a poor growing donor. The donor was positive for megakaryovirus virus when it arrived into our labs. And in our standard bioreactor and T-flask process, we were generating somewhere between 2 and 3 million cells per milliliter over a 7-day process. Not ideal if you're trying to develop a high yield product for autologous CAR-T. However, taking that exact same donor material and applying that process control strategy, which dynamically feeds in certain reagents or nutrients based on certain measurements, we're able to take that exact same donor and generate effectively 4 times the number of cells, so 8 times 10 to the 6 cells we're able to generate, and again, in a shorter period of time. So down to about 5 days rather than 2 million cells over a 7-day process. So we're starting to show that actually you can generate significant yield improvements and potentially quality improvements by adapting the process based on the donor material needs. And this is work that we've shown to the FDA, we've shown to European and UK regulators, because they need to be part of this conversation. And they are very keen. I mean, they're not there to kind of stop everything coming forward. They're very keen to understand what are these algorithms, what are these process control strategies that they need to be thinking about, and to what extent, would it be allowed within a regulatory framework? And all we're arguing effectively is we're just extending the design space. We have a better understanding of our product, and we believe that we should be able to accommodate the patient variability and adapt the process clearly within a certain defined field, but in a way that is dynamic and can adapt to the patient material. So the final bit of data I want to show is, does establishing a perfusion process improve CAR-T yield? And again, the answer very much is yes. So this is data that's been generated by two of my doctoral students, Tiffany Hood and Pierre Sprinwell, looking at the production of T cells and CAR-T cells in perfusion bioreactors. And this is part of a large DOE study, which we've now finally finished, which is great. We have huge amounts of data. And again, we're looking to publish this work within the next kind of 2 to 3 months. We've done about 40 different bioreactor runs to generate a wide design space of key perfusion parameters to look at the impact of key parameters on cell production. And we've been able to show that actually we can generate very, very high densities we've never seen before in a lot of these cultures, up to 20 million cells per milliliter, in some cases actually higher. And it's fascinating to see just how far we can push these cells in terms of the production. Now, this has importance for 2 reasons. From a patient standpoint, they don't need billions and billions of cells if it's a patient-specific process. They just need, maybe 500 to 1 billion cells. That's a normal dose for CAR-T products. But this starts to allow us to think, can we generate those cells in a shorter period of time? So instead of generating that billion cells over 7 days, can we do it in 24, 48 hours? But from an allogeneic standpoint, where we take healthy donor material and generate CAR-T cells from that to give to maybe 100 or 1,000 patients, obviously this has significant importance because we can show we can push the cells and generate significant numbers and therefore higher doses. The final part of my talk, and I'll kind of touch on this briefly, and this is really work that's been led by my colleagues, Dr. Stephen Goldrick and Dr. Joe Egan, is the process modeling AI and digital twin for cell and gene therapies. So this is part of a 10 million euro project, working with a number of our European collaborators, similar collaborators to our AutoSTEM project, such as IPT Fraunhofer. looking at what we call the ATMP project, artificial intelligence-driven decentralized production for advanced therapies in the hospital. So the idea of developing a completely automated platform that can sit within a hospital setting that is driven in part by AI digital twins, as well as advanced bioprocess control capability to deliver a non-viral CAR-T process and product for patients that require CAR-T therapies. Now, this is in partnership with the University of Würzburg Hospital. So the platform, and we're finishing the project this year, the platform will be in situ at the hospital in the next kind of 4 months or so. And then we're going to be running validation runs for the remainder of the project to demonstrate the impact and the outcome of the system itself. So as I mentioned, this is a non-viral process, and I'll touch on that very briefly. But the idea, so the key work at UCL is twofold. The bioprocessing aspects and the kind of non-viral bioprocessing and generating data. But ultimately, the second part, which is led by Stephen and Joe, focused on the bioprocess and the digital twin aspects. So as part of our high-throughput bioreactor studies, we've done lots of studies in the stirred tanks, in the Lonza cocoon platform, in the G-Rex bioreactors, and one of the partners in the project, the Aglaris, a company who have developed the FACER system, which I won't go into the nuances of the differences between these platforms. Happy to have a conversation about that over drinks. But in terms of some of the key outputs, this is actually more of the data from those high throughput perfusion studies. That's I think that's all 36 runs that we have there. We've been able to show through our DOE approaches, the importance of key parameters and kind of where perhaps the sweet spot lies in certain cases. We've moved that, as I mentioned earlier, to the ½ liter to 1 liter and now 2 liter studies. And we started to look at the other platforms that could be used for CAR-T production, such as the Lonza cocoon system, and developing a completely non-viral workflow, which I'll touch on the next slide. And here's some of the data we've generated with the Amber 250 and 2 liter stirred tank bioreactors for CAR-T production. So as I mentioned very quickly, we are looking at developing a non-viral workflow for CAR-T production, and that's kind of looking at a completely closed workflow using the Lonza nuclear effector, connecting that to the Lonza cocoon, and we've done some studies to optimize the electroporation process as well as generate expansion in the relevant systems. The final bit I wanted to show, and this is actually work, and I want to make sure I effectively signpost by Stephen and Joe, who are part of the project, is looking at how we can utilize some of those data sets to generate a online real-time digital twin. So how can we mimic the process and kind of estimate or guesstimate or predict the outcomes from the process whilst it's running based on real-time measurements and readings. And so as part of this, they, based on data that we've generated, but also some of the industrial partners have generated, have developed a digital twin, which looks at how we can estimate the production of CAR-T. So I won't go through all the nuances just now, but if you look at this here, this is a brief animation. The red line, or the red dotted line, is the estimated prediction from the digital twin, and the blue dotted line is what actually happened. Obviously, we didn't know what was going to happen, but this has been put together retrospectively. So when you look at the first kind of 24, 48 hours, there's quite a big distance between the predicted model versus the actual outcome. But as the model gets more and more data, as the process is running in real time, you'll notice that these lines tend to get much, much closer. And we're starting to generate, through some of this work, some real-time online predictions of a bioprocess. And again, from a patient standpoint, that is absolutely critical because every patient will be different. So therefore, can we really get an understanding as the process is running, what is going to happen in 3 or 4 days, or when can we harvest, when can we make some of these key inflection points? And that's important from a scheduling activity. Patients who are needing CAR-T are suffering from certain forms of cancer in many cases, which is aggressive. It debilitates them. Even going to hospitals is a major challenge. So being able to schedule when the patients need to come in with accuracy is critical. In addition to, obviously, do we know if the cells are going to generate enough cells the process is going to generate enough cells for a single dose? In many cases, Novartis have shown they've run through a 15-day process, and at the end of it, they've had to terminate because they couldn't generate enough cells to treat the patient. That's 15 days wasted. That's a whole process run, and they're not being able to generate any meaningful outcome. But most importantly for the patient, it means they've not been able to get a CAR-T therapy. And are there things that we can do to improve the outcome, but also predict the outcome as well? So I'd like to finish with this final point. Manufacturing is only half the problem, particularly bioprocessing manufacturing is only half the problem. There's a number of other problems, but I think one of the critical challenges is skills. And the Cell and Gene Therapy Catapult have highlighted in their various skills reports the fact that we have a number of companies, organizations that are trying to recruit bioprocessing experts in particular, and there is a major, major shortage. This was actually their 2019 data, and I deliberately show this because they've got their 2024 forecast, and we're actually well above the number of total jobs that are required, and the number of bioprocessing roles that will need to be filled, and we're not keeping up with the targets. We're not able to generate enough people, trained individuals, who can support the sector. And so what are we trying to do to address that at UCL? We've got a number of activities within the cell and gene therapy space, right the way from our undergraduate programs, where we have regenerative medicine minor, focusing exclusively on advanced therapies, and that is applicable and available to all engineering students, whether they're from computer science, from chemical engineering, mechanical engineering, and obviously biochemical engineering. And that's a very popular minor across the minor program. We've also launched a new MSc program called the Manufacture and Commercialization of Stem Cell and Gene Therapies. I think it's the longest MSc title at UCL, and I take the blame for that, but we want to capture keywords within the we want to make it clear this isn't a life sciences MSc. This is not an MSc focused on stem cell biology or gene therapy, immunology or virology or whatever. This is focused very much on the translation, manufacture, and commercialization. How do we address that challenge of making this sector viable? We've also got a number of doctorate programs. I'm delighted to be partnering with Cytiva as part of the UCL-Cytiva Center of Excellence. And we have a number of post-experience activities. And one of the key ones is the MBI in cell and gene therapy, which is actually running from the 4th to the 7th of March, happens annually. So in a couple of months, 4th to the 7th of March, we'll be running the next iteration of that in person at our new UCL East campus. So if you would like to join those online or in person, by all means, please do so. But I want to mention very briefly our MSc in Manufacturing and Commercialization, because the focus is to generate future leaders, those that have the scientific and technical understanding, but also those that have a focus on the business and commercial aspects. Since 2023, so September 2023, we've delivered this at our new UCL East campus. And also I wanted to mention this because, again, Cytiva have been instrumental in supporting some of this training activity. And since the program's inception, have awarded 2 $10,000 scholarships to eligible students on that particular program. So again, many thanks to our colleagues at Cytiva for their support. So to wrap up, I think from my perspective, we have to generate improved process understanding and look at how we can employ automated technologies, whether that be robotic platforms or improved automated process control to facilitate scale-up and translation. We have to look at how we can utilize process control and employ digital tools such as AI and machine learning as part of that. Obviously, we're trying to develop scalable and control manufacturing platforms, and ultimately we have to improve and enhance our analytical platforms to accommodate some of the challenges with the data we're generating and demonstrating efficacy. So with that, I'd like to thank a number of colleagues. So within UCL, obviously all my academic colleagues, but also in particular my research group have been fantastic. A number of individuals who have supported some of the work and their data have demonstrated. A number of colleagues at Cytiva who have either directly supervised projects or been involved in setting some of the aims and objectives and scientific rationale for the projects. And here you can see some of the various images and key people as part of the project as well. So with that, I'd like to thank you all for your attention and happy to take any questions.